Epigenetics is the study of the mechanisms that allow the inheritance of different gene expression profiles in the presence of the same DNA sequence. The genome is defined as the set of genetic material and therefore the entire DNA sequence contained in a cell or organism. A new study reveals that cancer can develop purely from epigenetic changes, challenging the conventional belief that genetic mutations are necessary for the disease. Scientists focused on epigenetic factors called polycomb proteins, which regulate the expression of key genes and are dysregulated in many human cancers. When these proteins are experimentally removed, the activity of the targeted genes is disrupted. Some can activate their own transcription and self-maintain. When polycomb proteins are integrated back into the cell, a subset of the genes are resistant to the proteins and remain dysregulated through cell division, allowing the cancer to continue its progression. Researchers find that brief and reversible inhibition of a gene silencing mechanism leads to irreversible tumor formation in fruit flies, challenging the idea that cancer is caused only by permanent changes to DNA. The formation of tumors and progression to cancer are usually thought to be driven by the accumulation of permanent genetic mutations. Specific genetic mutations that have been linked to cancer often alter gene expression programs, promoting changes in a host of cellular functions, including proliferation, differentiation, metabolism, and survival. Although cancer initiation and progression are generally associated with the accumulation of somatic mutations, substantial epigenomic alterations underlie many aspects of tumorigenesis and cancer susceptibility, suggesting that genetic mechanisms might not be the only drivers of malignant transformation. However, whether purely non-genetic mechanisms are sufficient to initiate tumorigenesis irrespective of mutations has been unknown. A new study has shown that a transient perturbation of transcriptional silencing mediated by polycomb group proteins is sufficient to induce an irreversible switch to a cancer cell fate in Drosophila. Polycomb group PCG proteins are epigenetic factors, forming two main classes of complexes called polycomb repressive complex 1 and 2, which are highly conserved from fly to human and play a critical role in cellular memory by repressing developmental genes throughout development. PCG dysregulation leads to cell fate changes, developmental transformations, and is associated with cancer. A research team, including scientists from the Institut de Génétique Humaine CNRS Université de Montpellier, has discovered that cancer, one of the leading causes of death worldwide, can be caused entirely by epigenetic changes. In other words, changes that contribute to how gene expression is regulated and partly explain why, despite an identical genome, an individual develops very different cells, neurons, skin cells, etc. While studies have already described the influence of these processes in the development of cancer, this is the first time that scientists have demonstrated that genetic mutations are not essential for the onset of the disease. This discovery forces us to reconsider the theory that, for more than 30 years, has assumed that cancers are predominantly genetic diseases caused necessarily by DNA mutations that accumulate at the genome level. To show this, the research team focused on epigenetic factors that can alter gene activity. By causing epigenetic dysregulation in Drosophila and then restoring the cells to their normal state, scientists have found that part of the genome remains dysfunctional. This phenomenon induces a tumor state that is maintained autonomously and continues to progress, keeping in memory the cancerous status of these cells, even though the signal that caused it has been restored. Thank you for watching. If you've enjoyed our videos, subscribe to our channel for there's more to come.